My name is Clinton. My question has to do with your comments on the impotence of natural selection. Now that gene sequencing has gotten reasonably cheap, someone did a group of chimps, and they found that the infant chimps had 35 differences on average in their DNA between that of either of their parents. Now, it seems to me that if you got 36 differences in an individual from his parents, that natural selection has something to get a bite on.
That's exactly natural selection. And natural selection is very powerful. But those 35 differences come from rearrangements of the existing genes in both parents. I mean, we take half the genes we've got, and we make sex cells, and then we take half of what we got, shuffle the deck, and deal out a hand to the next generation. But you can shuffle and deal all you want, but monkeys only have monkey DNA, monkey genes to give. What we see as differences, like you and I look different from our parents and our grandparents, but we also look similar. What we've got is we've been dealt from a deck just a different selection, arrangement, a combination of pre-existing genes. And I didn't say that natural selection was impotent. It is a very powerful force, but it is in no sense a creative force. It's only a force that conserves the integrity of a gene pool.
Than that, and gene doubling is quite a significant addition to anybody's gene pool.
And gene doubling, isn't that just making an extra copy of a pre-existing gene?
That's something else that gets worked on.
Okay, now there's where the theory comes in. The fact is, a gene can get doubled in a living thing. And the answer always is that extra gene then, while the old one, the original one, can continue doing its job with fidelity, the new one now becomes the tinkering workshop for evolution. Now, how evolution can actually tinker and deliberately, and Stephen Jay Gould roll over in his grave at any evolution that says anything's happening deliberately, how it could actually then tinker with this extra gene until it makes something brand new with new genetic information. The gene doubling, we can see, but actually the gene that gets doubled and then changed into something better, that we don't see. Actually, Gould's left wall hypothesis says it only looks like things are getting better, and he totally believes in evolution. The fact of the matter is, when you start out as bacteria, it's like a drunk guy walking along a brick wall. He called this the left wall hypothesis, that he bounces off the wall, he gets further from the wall, he appears to get further in the right direction, but that's only because there's no place left to go but up when you're a bacterium. And so it's only, evolution gives the illusion that it's trying to make things better. And this is Gould's hypothesis. And I think that's very true. If I believed in evolution, I would recognize that as valid. But gene doubling, we see it happening, but then actually the doubled gene becoming something bigger and better is not, that's something we actually don't see happening. It's a great idea if you believe in evolution, and it'd be a nice evolutionary posit, but we don't see that part. And that's where I don't want to even call it theoretical, that's where the evolution story comes in, which is okay, and it's something that could happen, but science has to go on documentation.
Hi, Mark. The point of your talk seems to be evolution is not science, creation is not science. In the middle, or creation theory is not science. In the middle of your talk, you talked about hard science versus soft science, and maybe there's other forms of inquiry, maybe that's philosophy. Would you mind filling in what distinguishes hard science from soft science and maybe other forms of inquiry, and then give us some examples of what's hard science and what's soft science?
Sure. I mean, Albert Einstein said imagination is more important than information, and that's true. You have to dream up these scenarios, thought experiments like he did, and that's very, very important, but it's soft science until you actually have some observations. I mean, even if you've got a brilliant idea that's right, like E equals mc squared, like the gravity can bend starlight, he had to test that out during the first solar eclipse where there wasn't a cloudy day, and test it out. So hard science would be data-driven. You can subject it to the scientific method, hypothesis, the question, the hypothesis, the procedure, an experiment with controlled and controlled variables and experimental variables, all of that. and then data collection, observations, analysis, and then give a conclusion that is based on those things. That's hard science. The scientific method as devised by Sir Francis Bacon. Soft science would be hypothesizing, brainstorming, or things that you got enough things that hint that your idea is right, that it's kind of scintillating, and people are interested in it. And ooh, my gosh, this guy could be right, you know. But you don't get the Nobel Prize for soft science unless it's in philosophy. So I would separate hard science from soft science as anything that's observational science, that's data gathered. It's evidence, it's observations, measurements. You can quantify it. That would be hard science. And other things are things that are cool, like talking about what the life forms might look like on around Gliese 581. You know, if there's that planet that's going around that star, maybe there's life there. What would it maybe look like? You know, that's soft science because there's no way to observe it. Astrobiology is completely soft science. There's no data. They have to make up stuff to study and talk about. But it's not like there's no value in that. There is value in it, but it isn't hard science. Does that help?
Maybe, what about disciplines? Like, what you mentioned geology, tectonic plates. What is geology, a hard science or a soft science?
Well, I suppose that Alfred Wegener's theory was soft science until they actually found the magnetic striping on the sea floor in the 1960s. And he happened to be right. His soft science was able to make the jump to hard science because it was confirmed by observations. I'm sorry now, what was your, I went with it. What was your question again?
Examples of hard science versus soft science. So geology.
Disciplines. Yeah, certainly, and no joke about rock hard science and geology, but anything in geology that is testable, like, you know, drop some weak acid solution on a calcite crystal and watch it fizz, positive test for calcium carbonate being present. You know, but if you're then talking about the geological composition of the planetoid Sedna, we just don't know. Now, it's fun to talk about, but you can't do any geology with Sedna. We've barely gotten to where we can do geology with with the Moon and Mars and Venus, and it's limited. We don't do any with Jupiter. Does that help you see if you got actual data?
I think so. Just one last example. What about just astronomy?
Well, most
science.
of my examples were just from astronomy. Is astronomy a hard science? Sure. Light beams coming through the lenses of a telescope, radio waves coming through a radio telescope. But talking about what the aliens look like on some other planet, that's got to be soft science. Any data that you can actually gather. And if you don't believe in your senses, if you don't believe optics, you don't believe in light waves, you don't believe that what we're seeing is really true, then you can't be sure about anything. So we're just going to have to go on that our senses aren't lying to us, that we actually do see the light, you know, from Alpha Proxima in our telescopes, and we can do the spectral analysis of the light beams. The observational senses are what we're all trusting is real and makes it hard. But so astronomy's got lots of hard science, but it's got lots of, you know, fluffy science too. But for many people in science, that's what makes it fun, is the fun imaginative stuff. But we don't sell soft science as hard science, and we don't get Nobel Prizes for that either.
Hi, my name is Max Lyon. So you said before that anytime a scientist comes out with something that they propose as a missing link to evolution, they always put the caveat on there that it's not the missing link or that the missing link is still out there. Just from your point, or from your position, what would the missing link, I guess as you're putting it, be? Or what would it have to look like?
Well, we've got missing links between the prokaryotes and eukaryotes. The biggest jump is from non-life to life. Prokaryotes to eukaryotes. Evolutionists have Lynn Margulis to thank for her theory of symbionts. But there's not just one missing link. I assume you're talking from monkey-like creatures to humans. Is that what you're talking about?
I think that was the example you gave.
In that one, Well, of course, it would have to be in the right layer to correspond with evolutionary dating of the layer. It would have to come at the right part in the hypothesized story of us splitting off from the monkeys genetically 6 million years ago, or fossil-wise 7 million years ago. Sahelanthropus chilensis has kind of been rejected because it's 7 million years old, and the genetic data is interpreted as 6 million years ago it would have happened. It would have to actually fit the evolutionists' critique and characterization of where we're supposed to find it in the layers and what it's supposed to look like, how far along the way from getting from a simian-like creature to Homo, our genus. It would be determined by evolutionists themselves, because I don't believe in evolution. To me, I study it like I study the characters in Lord of the Rings. That's how I feel about it. I have to. I don't think any of it's true. So I'm not saying there aren't a bunch of fossils, but I'm saying, as you might have saw the placard earlier, there are plenty of monkey fossils, plenty of human fossils, but no monkey humans. I don't know, did you have one in mind in particular?
Well, just so I would assume then you would say that any of the fossils that have been proposed as intermediates between then were falsified or were just wrong?
Well, there's usually a bone structure thing is wrong. It's in the wrong layer, like Australopithecus africanus is actually in the wrong layer, and so it makes the story go backwards for 1 million years. And that would bother me if I was an evolutionist. Or the person who dug it up, like Tim White dug up Ardipithecus, says it's not the missing link. The team that dug up the Ichthyostega and the Acanthostega, they said that these things couldn't manage to do more than flop around on land. That's the missing link between fish and amphibians, between ocean creatures and land-walking creatures. And I've been to the Sam Noble Museum of Earth History, and they've got a lie on display. Every single fossil of Ichthyostega has 7 bones in the paddles. It was Michael Coates and Jennifer Clack who dug up those fossils, and they said their limbs were pretty much paddles. They couldn't have managed to have done much more than flop around on land. But the museum has this thing as this big, sort of Komodo dragon thing, and all the drawings and textbooks and all the models I've seen in museums have 5 toes on these muscular legs. The people who dug up the fossils in 1999 said their limbs were paddles, and every Ichthyostegid fossil has 7 bones in the paddle. Every Acanthostega has 8. Well, they got that part right. But that frustrates me, that what everybody in paleontology knows isn't applied to the models that are set up in the museum, because all land-based vertebrates have a system, a standard of 5 digits. And so they thought that Ichthyostega would, and when they discovered it did not, they didn't change anything in the books, and that bothers me. So yeah, there's usually something that disqualifies it, or the person says, guys, let's keep looking, it's pretty interesting, but it isn't the missing link. Yeah, and if you actually had picked one of the hominid, things, I could have told you what the problems with it are, and if I was an evolutionist, I would keep it all as tentative and just keep looking. Yeah.
Hi, my name is Ryan. Thank you so much for your talk. We appreciate it. I have a kind of a 3-part question. The first one's very simple. It's going to seem a little unrelated, but how long have you been a Christian creationist?
Well, let's see.
Let's separate this two. How long have you been a Christian? How long have you been a creationist?
Do you really want to know?
Absolutely.
Okay, because here it comes. Now you're going to get into something theological.
Sure, a little
because,
bit.
you asked about my religious beliefs. Now, I heard Eugenie Scott, head of the National Center for Science Education, she's got a doctorate in paleontology and she's an atheist. I heard her say that 70% of Americans identify themselves as Christians. 30% of those identify themselves as born-again Christians. She said, now they're the ones that we evolutionists can't work with. They just won't listen. Those born-again Christians. And then she defined, I was right there in the audience at University of Cincinnati. She said, now born-again Christians, she said, they're the ones that have Jesus Christ living in their hearts. She didn't even say think Jesus lives in there. She said they're the ones that, and I'm like, my goodness, she knows. And so in my religious belief, I believe there is a God, and yeah, that Jesus lives in my heart. I got saved when I was in senior in high school. Now, I was 17, but I was already totally a Sheldon Cooper science geek. My mother worked for NASA as a secretary. She took dictation from Carl Sagan once. When I was 6, we would watch on TV the Friendship capsule and the Mercury capsule launches, and my mom would say, "There's my boss, Dr. Ladair, down at NASA." There's the NASA guy, yeah. And so they knew that she had a little boy at home who loved space, and I got all these glossy pictures of the moon and all these other things and little models of rockets and a map of the solar system I memorized. So I was a total science geek. When I was in 7th grade, I thought I'd discovered parallel circuitry. Someone else really beat me to it. I was 9, I was raising land snails. They were laying eggs and hatching babies. I had a chemistry set, a telescope and a microscope, little electronics set. I was playing with wires and building my own little electrical things, and it was a lot of fun for me. And I made a Babbage device from a kit, which is kind of like a Turing machine, but it was a little teeny thing made of plastic, and it just could multiply, add, divide, and subtract. So I was like a total science geek. My grandfather gave me a book called The Boy Scientist. So what I'm trying to say to you, by the time I was 17, I knew I wanted to be a science teacher. I totally believed in evolution. Yep, the day I had my conversion to Christianity, and I did not expect this to happen, it was like I'd gone to Mos Eisley and had a memory wipe or something. I mean, not that I was brainwashed by becoming a Christian, but my whole perspective, it's like everything lifted up out of those little pegs they were in, rearranged, and then got reset down. I went, oh my gosh, evolution can't be true. I actually instantly that day didn't believe evolution anymore. I didn't know why. I couldn't prove why. So I started studying these kinds of things that I've shown you. So there's a religious idea. And so I became a Christian first, but it was almost simultaneous that I quit believing in evolution. No one actually pressured me to do that. There were people in my church who believed in evolution. Lots of Christians believe in evolution. It's called theistic evolution. Francis Collins, the head of the NIH, he grew up in the town my wife and I met in, and we got to talk with him. He is a Christian who believes in evolution. Mary Schweitzer is a Christian who believes in evolution, who found the red blood cells in the T-Rex. So it's a theological issue, but it does impact my worldview, which impacts how I'm going to interpret data, because now I have a different paradigm. And that happened when I was 17, but I was already pretty inculcated.
Sure. So kind of my main question was that obviously your theological view has influenced your scientific view, whether it's historical
everyone's
science.
does. It doesn't matter what your theological view. Yeah.
I disagree with that. I really
do.
do.
You do?
I do.
Well, it does. Okay, well, let's get personal with you then. For your theological view, are you saying it doesn't affect your scientific views?
I do not think so. For myself personally, no.
What is your
historical science?
What is your theological view?
My theological view, as of right now, is atheist. I grew up Christian. I was the worship leader in my church for 4 years. At that time, honestly, I didn't really have a position on evolution because I didn't think it was a positive position to take. During your presentation, you talked a lot about how you don't like Richard Dawkins, how you don't think he's...
Well,
you
I
respect
personally
him,
have
but
a
you had
problem.
3 quotes from Richard Dawkins in your presentation. You talked about how you do have respect for Neil deGrasse Tyson. I didn't see one quote from Neil deGrasse Tyson on evolution. You had a quote from Lori Olem-Weinstein, who's a psychologist on evolution. You had quotes from National Geographic, from high school textbooks that honestly don't depict an accurate depiction of evolution.
Hold
sorry.
on a second.
Sure.
In 45 minutes, I wasn't going to cover all the bases, and I certainly wasn't going to be able to do a completely even-handed presentation. The point was to make the point of whether evolution is scientific or not. Now let's back up again. You actually are going to state that atheism has no impact on the way you view science. Your atheism. Your brand of atheism. Okay, now you have a very actual, customized atheistic position, true?
Yes.
Yes, and you are actually going to say that that does not color any of your perceptions about reality in the slightest.
Not my perceptions of reality, my perceptions of historical scientific evolution.
Oh, well, of course, so it's going to affect, because creation is a historical scientific perspective, evolution is a historical science perspective. So because of your atheism, you choose the evolutionary historical science perspective, right?
No, because of the evidence, I choose the historical scientific evolution.
Well, like what?
How long was your presentation?
Yeah,
45
well, just
minutes. I don't
pick
have 45
one.
minutes to go
Just
through the
pick your
evidence. I mean,
favorite.
the fossil record is a great one.
Well, which fossil is the
DNA
best?
I don't have 45 minutes. I mean,
No,
if you
just
want
tell me
45 minutes,
one.
we can
That's
put
all.
our-
We're just one.
side by side. I mean, I'm more than happy to do that. I'm not an evolutionary biologist. I go with,
Well, then what
The
does
evidence
convince
for
you if it isn't the fossils?
sorry,
What does convince you if it isn't the fossils?
the DNA evidence, the evidence through lineage DNA.
Okay.
Like the evidence that we brought up earlier with the point of that was that each individual piece of evidence for the apes was unique to its mother and father. It wasn't just a reassimilation of DNA. The lineage of DNA throughout evolutionary history is one of the strongest pieces of evidence.
So
There's
you're
not,
saying
I don't think
that
there's
the
one piece.
offspring aren't really just a reassembling of the parent's DNA?
The DNA genetic sequencing wasn't just necessarily a random offspring.
Of random offspring.
Whenever the experiment that he was talking about wasn't just a normal genetic offspring. It wasn't just a mother and a father. It was genetic sequencing. That's different than just genetic offspring.
Okay, so they sequenced the DNA in some individual
The
chimps,
DNA
right?
that they were able to sequence from the mother and father was unique in
Of course.
within 35, and not just a reshuffling of individual DNA. It was the DNA itself was unique.
Well, of course, everyone in the room has unique DNA. I'm not sure what you mean.
The point is that throughout DNA lineage, the sequences are extremely similar, but they're also different. I mean, again, I don't have 45 minutes to go through the DNA evidence of DNA. I really don't.
Well, I did have 45 minutes, but now we've got 45 more minutes if we need it. What I'm trying to say is, it would fit with an evolutionary model, but it isn't the only way the observations we see about the DNA could happen. It could happen if creation was true, too. You're saying that the DNA of the parents is unique. Of course it is. And then it reshuffles and ends up in the kids, the offspring, and they are unique, too. Each person is not, unless you have an identical twin, and I guess those two twins are not identical, they're fraternal, there isn't anybody else with your exact genetic makeup unless you have an identical twin or a clone of you. So I'm not sure how uniqueness of an individual's genome is evidence that evolution must have done that.
The similarities are more evidence than the similarities between humans
Well,
and apes.
if
The similarities between
actually humans were created by God and we're all descended from the same original pair, and even in evolutionary genetic theory, we've got mitochondrial Eve and Y chromosome Adam, and they just recently came out that all people with blue eyes are descended from one individual. This was last month. One individual. All blue-eyed humans are descended from one individual who lived 6,000 years ago. Well, of course, I like that with the Bible time scale, but we actually find similarities because we all are common descent. Now, I don't believe we're common descent from monkeys, but humans are all common descent from original humans. And again, that could happen, the similarities between individuals could happen a lot of different ways, and evolution would be one of them, but it's not exclusive. In science, you have to find something that wouldn't be true unless your theory was true and no other way. And then we call it proof.
Sure. One last broad thing. I'd be happy to discuss different examples with you later on. I don't want to take up everyone's time, but
Yeah, but this is
obviously,
fun.
yeah, it is. Obviously, your religion colored your opinion of.
And you're still maintaining yours does not.
Yes, absolutely. 100%.
Okay, now you heard that. You heard that because religion is, of course, going to it's going to cause how you interpret all things, and I just can't see. I don't believe you're lying, but I also don't think you've thought this through. Your view of why the universe is here and how it got here has got to affect everything about your perspective, including on data itself. So anyway, I'll just agree to disagree with you on that
Thank you.
one then.
Thank you.
Okay, you're welcome, and thank you.
Hello and good evening, Doctor. My name is Stephen. Now I'm not anything like a biologist or a chemist. I'm a computer scientist, and unfortunately I'm not also an information theoretician for this next question. Now when you talk about information, is this, you talked a bit about duplication and there not seeming to be much in the way of new information when for example, mutations occur and things like that. But if a mutation is just something that causes any kind of measurable change in how that code is evaluated, the evaluation being the proteinic patterns that make up the organism, then where does the distinction come between this like older kind of reshuffled information and actual new information that would have presumably come either through creation events or a spontaneous, I guess you would call it an evolution event, although I'm not really sure that there's actually a presumption of this kind of information building in evolution.
Well, what I think is the crux of your comments is that the idea that mutations can actually, which are copying errors, it's like code being copied and downloaded, but an error is made in the copying of the code. Now, your DNA codes are very complex. You have 3 billion base pairs in every one of your cells. So we're talking about a lot of bits and bytes, okay, of information. And the idea that such a complex code could make a copying error and actually improve and create new traits. For instance, that bacteria became humans. Now, oh yeah, give it 1.5 billion years and it maybe could happen if you believe that the longer you wait, the more possible the impossible is. But that's what you got. You got to start out with bacterial DNA and then it's got to work its way up, you know, to us. Now, just by copying mistakes, if you make a copying error, it's not directional, and natural selection can winnow out good and bad if good and bad arrive, but it can't actually write new information. To think that copying errors could actually write new information that is useful never comes from any place but a mind, and there's no imaginable example where that isn't true. Usable information, the kind of information in your DNA, it's a recipe on how to make something. It's instructions on how to do something, how to assemble these proteins that you were talking about. And we now know that there are 22,000 functional genes, and some of them actually code for more than one thing. Most of them do. Now we found out if you read the gene backwards, it codes for another useful protein. And there are actually control systems that will take pieces from many different genes and then as much as 38 of them on different chromosomes and splice that code together in a messenger RNA, send it out to the endoplasmic reticulum. This means that it's far, far more complicated. Information nested within information. We're not just talking gene methylation here. We're talking cut and splice an entire system. This is how 22,000 genes can code for over 1 million proteins by all of this cut and splice, and it's all coordinated by the junk DNA, which is actually turns out to be the master regulatory genes, the central nervous system of the genome, that 98% that, since we're so smart and we couldn't figure out what's doing, it must not be doing anything, it is controlling all of that. So what I'm trying to say is, you change one thing, like the gene that controls hearing, and the gene that controls the healing process of cuts is the same gene. You mess with that gene to give you super hearing, and now your cuts don't heal. You just random haphazard. And if mutations are so good, why do you have secondary backup systems and repair mechanisms and editing enzymes that repair any mutation that might happen? Your body systems are totally geared for stopping mutations. We mutated, I think it was field daisies or black-eyed Susans, I forget which, but we mutated that. After 3 generations, 3 generations, the mutation was reversed and healed. The generations actually reversed it. Now, in your own body, you have editing enzymes that roll up and down your DNA strands, and if they find a nucleotide base that's out of the right order, it'll remove it, grab a spare part from the nucleoplasm, and insert it. And so you've got these backup systems and checks that actually everything in your genetic biology is geared for stopping these mutations. And if it's so wonderful, why don't we just go ahead and mutate at super speeds? And why don't bacteria, just turn into all kinds of things? It's wonderful. Biology is against actual mutation. And the idea that even if a mutation does get through, that it's going to make some kind of superior gene, new genetic information, new traits that are actually better and confer greater survivability in a new environment like an ice age or something, we actually don't ever see that happening. So I tried to keep it as non-biological as I could and appeal to your computer guy thing and try to stay away from information science of any super hardness.
I guess I'll try and clarify a little bit. There was an establishing point years back where they had to stop calling it junk DNA because
2012.
2012, thank you. And because there were beneficial, mechanisms in place with regards to how this extra genetic material was actually interacting with the rest of the organism, and because for various reasons it would acted as kind of a buffer. One of the examples was it acted as kind of a buffer in case a lot of genetic damage occurred. So from that you can establish that okay, there isn't really junk DNA, but there is DNA that doesn't actually reflect in the final form of the organism in any actual protein action. So now, as you know, like if you screw around with something, now this is a terrible analogy actually, because computers, we really don't have any computers that really behave like the evaluation of DNA into proteins. But if you screw around with like the instruction order in a working program, oftentimes you can cause jumps into uninitialized memory that contains a lot of junk, and it will still execute for a few cycles because that junk data may actually be interpreted as valid instructions for a little while. It's all part of the same set of symbols. In this case, the GCTA, I can't remember the actual names of the nucleotides. So when you say that there is no new information to be had in organisms, now we've established that even though mutations are usually repaired, they can persist. And there is, if you happen into something that does not actually destroy the organism, are you saying that there's no way that difference can persist?
There are plenty of, you're carrying over 300 lethal recessive genes yourself, all of us are. It can persist if it's recessive and you've got a backup gene that can take care of it. And those are, of course, the most insidious of the genetic problems. Now, you did say that I had said that junk DNA, you said that junk DNA isn't actually contributing to the production of useful proteins. But I didn't actually say that, and that's not what the findings were. The shocking finding was, in 2005, 2007, all confirmed in 2012, was we can't actually find any sequences that aren't doing anything except mutated, broken down genes. We think that probably 100% of the genome is being transcribed, and that's a lot of energy for the cell to do for something that doesn't function. The first junk sequence we discovered that was doing something had to do with males and females. Females have 2 X chromosomes. Males, like me, only have 1. And actually, a double dose would be bad of the genes on the X chromosome. And so we knew that half the genes on one of the X chromosomes in a female will deactivate, and the other half will deactivate on the other one. And anything the female needs a double dose of, they're left operating on both X chromosomes. Now, we found out in 1999 there was a portion of the DNA that was in the junk sequence that was actually calling all the shots on this, was coordinating these activities. And so all of the junk sequences that aren't coding for a protein actually tell all the other gene coding sequences when to do it and how to do it and how much to do it. Another one is the FOXP2 gene that was experimented with by Svante Pabo at the Leipzig Institute the Max Planck Institute in Leipzig, Germany. And Pabo found out that the FOXP2 gene, which controls, among other things, brain development, produced a protein that was only 2 amino acids different in us from monkeys. And he knew that's not enough of a difference in the protein that controls brain development to make our brain 3 times bigger and us be able to talk. And so he was saying there's a lot more going on here than we thought. The FOXP2 gene must be cranking out a whole lot more of the protein in a human and at different times in our life cycle than in chimps. Normally a protein is 400 or 500 amino acids long, so just 2 amino acids different really shouldn't make that much of a difference in us. So that's another thing we began to realize something else deeper was going on. Some control system, which is in the DNA, is calling all the shots of when the other genes activate, when they get their chromosomal puffs, and how much they produce and how they do it, even determining the folding of the proteins after they're produced. And even the foldings in the DNA strands may encode information, layers upon layers upon layers. So I wasn't sure what your question was, but one thing I wanted to make sure everyone knew was that there really, what we've discovered, the shocking thing that surprised everyone and delighted them because it was new information, creationists and evolutionists alike, was that none of the sequences, can we now say, aren't actually doing something that indirectly, we could have a gene that tells a gene that tells a gene that tells another gene that tells another gene that tells a gene how to make what it makes and when. These nested feedback loops and all, control things like when you start cranking the estrogen and testosterone at puberty, when puberty is going to start, gauging your nutrition and all the other kind of things. It's just these incredibly networked and codependent systems that are way more complicated. Just the vast interconnectedness of it is obvious that any change you make in one place is going to affect a whole lot of other things, and the risk you run of screwing up 10 other systems by a lucky mutation that might improve one is probably not got survival value. Now, most of the mutations that we are carrying are relatively neutral to our survival, or we wouldn't be able to survive. There are some that, when they express, are detrimental to our survival, like the broken gene for lactose digestion, the lactase gene. And you can live, you just shouldn't eat soft serve. You shouldn't eat an ice cream cone. What the point that creationists make on genetics is we don't actually document new genetic material that's useful, coding for new and superior traits. Even in bacteria, we don't see new. The nylon-eating bacteria is kind of a hodgepodge. It's a chimera of several things that did happen, creating a myth that bacteria decided to evolve because they needed to. There's really no one nylonase enzyme that the bacteria were making. It's a suite of about 5 different enzymes and caused by a point mutation, and isn't really the way that the spin is if you read the reports. So what I'm saying, though, is bacteria have genes that are so simple that if a mistake did happen, there's a higher chance that it might not hurt it, or it might actually do something new that still works. And I like to tell my classes, what if you have the word heel, H-E-E-L, Change it to H-E-A-L, and it sounds the same, but it does mean something different. Change it to H-E-L-L, and now it not only doesn't sound the same, you see, but you can fiddle with short words. You fiddle with a longer word, like disobedience, and change the words, the letters around. It's not going to make any sense. The simpler systems, if you tweak them by chance, random copying errors, there's a greater chance you might make it through and might even have a lateral change and might even make an improvement. But we still don't have any documentation of that ever happening that isn't refutable. And even if it does happen, it could conceivably happen statistically, but not in eukaryotic cells. That means everything but bacteria. So yeah, you got me going on one of my favorite topics, junk DNA.
My name is Lauren Van Steenberg. I have a question. When you talked about the early pteropods, why did you ignore the fossil Tiktaalik? It is the one pteropod that has one bone in the upper arm, 2 bones in the forearm, lots of bones in the wrist, and 5 digits.
You mean Tiktaalik rosei?
The one found by Neil Shubin.
Yeah, Neil Shubin, and also Jennifer Clack was involved in that. Ted Dashler went on the Colbert Report with a little model of the Tiktaalik rosei. It's an Inuit Eskimo word that means large freshwater fish. So even the word, the name, means that it's a fish. Now, Ted Dashler said it maybe even would have had lung tissue because this thing was supposed to be walking out on the land. Now, maybe even had lung tissue. He said that a couple of years later. They never found lung tissue, but he said it anyway. And when you actually look at the fossil, look at, hey, IT guy, go to missing links. Let's get a picture of a Tiktaalik fossil. Missing links, and how about you are not a fish? Go to that. You are in a fish, yeah. Okay, click on that, okay, and let's maximize that and click the little tic-tac-toe board. No, not that, back out. Okay, click the little tic-tac-toe board, that's good. And now maximize the window with the big square on the top by the X. Good. Okay, now scroll down, scroll down, scroll till we get to Tiktaalik. It's not doing it, there you go. Oh, there's Tiktaalik, okay, all right. And give me that one. Okay, and now make it like my PowerPoint so I have control of it. Thank you very much. Okay, now here is from Time magazine an artist's conception of Tiktaalik rosei, found in 2006 by Neil Shubin, Jennifer Clack, and all those, the little team in Ellesmere Island off the coast of Greenland. And they found it was the perfect fishapod. And look at that NBA basketball player leg there, and it certainly looks like it's a fish that could get up and walk out of the water. This is the fossil. Okay, this bothers me because I'm looking at it, and did it pull in like a turtle's neck or something? I'm not trying to make fun. I mean, this is the actual fossil. You have models in the museums. This is the Chicago Museum. And then there's a model of the skeleton, but the fossil actually looks like that. And that's what the artist's conception is of it. This does bother me because I think there's a little bit too much artistic license. They did find scales with the fossil. They never found the hindquarters. They found several, I think 9 different individuals. This creature really lived. But is it actually? The problem is it was dated at 382 million years ago, whereas other things, okay, here's the real problem. They found a fossil in, actually a footprint trail in a quarry in Poland that actually has a tail drag mark and very heavy footprints, and they said, 4-legged creatures' footprints force evolution rethink. It's older. It's 10 million years deeper in the layers than Tiktaalik. Tiktaalik can't be the ancestor of land-walking fish, because this thing, whatever it was, they drew the feet like that, but that's not the way the footprints look. There was something big walking around on the land 10 million years deeper in the fossil layers than Tiktaalik. So Tiktaalik is not the missing link to walking on the land. And Jennifer Clack admitted this. She said, "We thought we'd pin down the origin of limbed tetrapods, but we have to rethink the whole thing." That was ancient four-legged beasts leave their mark. Per Alberg, who also worked on Tiktaalik, said, "These results force us to reconsider the whole picture of the transition from fish to land animals." Now, these two people don't now not believe in evolution. They don't now not believe that we came from fish. Jennifer Clack said, humans are Sarcopterygian fish, that we are mutated fish. So she really believes that. But what they realize is that this new fossil data totally makes their story they thought was true about Tiktaalik just not true. So Tiktaalik is not a candidate for the missing link between water and land walking creatures, and all the people on the cutting edge of the research who believe in evolution have said, we got to redo this. And they're being honest. Now, they're not denying evolution. They're not even denying that we came from fish. They're just saying Tiktaalik doesn't work anymore.
Well, you showed one fossil limb. They actually found 1.5 Tiktaalicus, so they actually have 6 limbs. And some of those were much better than the one that you showed. But
so you are saying it's the missing link?
no, I'm saying that it was the one bone, 5 digits. I mean, where you were obviously ignoring that fact. But
well, no, I'm not ignoring things. I'm saying we can ignore it now because we've got something already 10 million years earlier than that walking around on the land. So this is not the ancestor. It can't be. Something else beat it to the land. They don't know what it is, and that's what they're looking for now. But they don't have a missing link anymore.
But that was not my real, I mean, I actually asked that because I heard the other question and really wanted that. There's a question I want to ask. And it's
Well, wait a minute. What was your question then?
The question I want to ask
Because I
is
thought
about,
you were putting out Tiktaalik as a very good evidence.
To point that out.
Wait a minute. Were you putting out Tiktaalik as the good evidence?
I was putting out Tiktaalicus. I was asking you why you omitted that from your earlier.
Well, I think it's obvious.
Okay.
Because it isn't a missing link that even evolutionists believe anymore.
My next question is a little bit more theology, but fortunately it has a little botany in it, or plant biology. And it's kind of going to be somewhat confusing because I have a verse out of the Bible that I want somebody to read. Would you mind reading a little scripture?
Oh, why don't you read it for me?
Or,
Maybe it's one I got memorized from Sunday school. I don't know. Which one is it? What verse is it?
The reason I want to go to this, I just got through reading your website, your statement of faith from the...
Which is my faith.
Right, and it says that the Bible is infallible and perfect.
Which is my opinion.
Yes, okay.
Yeah.
Well, fine.
I'm entitled. Go on.
Go on, fine. Would you go ahead and read that scripture and...
What verse is it?
It's Mark 4:21.
Okay, go ahead.
It is like a mustard seed, which when it is sown on the ground is smaller than all the seeds on the earth.
And my question for you, and I've got one other thing so that I kind of need to you go to a spice aisle. This is the botany part of it. How large is that seed in comparison to these other 2 seeds? I will tell you what they are. They're a mustard seed, a celery seed, and a poppy seed. Which one's the largest?
Sir, are you trying to point out small corners of the Bible where whatever it meant by mustard, you're going to say it was scientifically inaccurate? Is that what your point is?
The book, I mean, obviously...
So you're saying the Bible is scientifically inaccurate. Well, like the ark was supposed to be made of gopher wood, but no one really knows what gopher wood was. There's nothing we call that now. We do have a plant that 2,000 years later we call the mustard seed. I don't know, there's the mustard family, and there's a whole group of plants that are called mustards in science today. Now, what Jesus was referring to was for an illustration. It was one of his parable stories. I do not know for sure what kind of plant he was actually referring to. It said that some guy climbed up in a sycamine tree, which I guess is a sycamore, but I don't know if we still call it the same thing or if it was translated to the word we actually have today. I don't know what the Greek word is. If you're actually trying to say that the Bible is a bunky book full of fairy tales because it has errors in it, I think you're probably going to have to do a little better than that as far as I don't know what your point is. That the point you're trying to make?
Yeah, but what I was trying to point out that the Bible is not an infallible book. I mean, I'm sorry. I think mustard seed was probably the same mustard seed he was talking about back
You
then.
think it is, and you're entitled to your opinion too. Thank you. Yes.
Hi, my name is Marcus Wiles. There was an article I read last semester in my evolution class. It was talking about salamanders, and we were also talking about segregation, how descent through modification, evolution, all that fun stuff. My question's at the end of the statement, so bear with me.
Is this going to be about ring speciation?
Essentially, how species diverge, go through physical barriers. They change along the way of the physical barriers. They can no longer physically reproduce. Not that they don't want to, they won't. And genetically, they do not produce viable offspring, even though they're basically from the same genetic line.
Okay.
My real question is, you say there's no evolution. What do you call that, if not evolution?
Now, okay, the inability to reproduce, infertility, probably caused by the separation of these two populations. Different mutations arise in these different populations, and their coping strategies genetically are going to be different. And you try to rebreed them together, if it hasn't been very long, they should still be able to rebreed like the mule and the donkey. I'm sorry, the donkey and the horse make a mule. And the lion and the tiger make a liger. I mean, that's really true. But there are things that have got, now there's mechanical reasons why timber wolves and chihuahuas don't mate, but that they won't, like you said. But actually saying that the loss of ability to be infertile is proof of this ongoing onward and upward evolution thing is kind of backwards. I mean, wouldn't they actually become better, more fertile as a species? Or you're trying to say that loss of ability to interbreed is proof of speciation?
More or less, yes. But it's not that they can't breed. They can breed along their own lines, but they're still basically coming from the same species, right? And through whatever it is, through genetic drift, essentially, they can no longer reproduce, even though they're close to the same species.
Okay,
I'm trying to find out, why is that not evolution?
Two populations that can no longer reproduce. You could do this with every species gene pool on the planet, and you're actually not getting better organisms. Don't forget, evolution in the Darwinian sense is, grandeur in this view of life. It's supposed to be an improvement. It's supposed to be a greater complexity and greater fitness. But all that we see in the salamanders being separated, this is ring speciation, they call it. The warblers on the Tibetan plateau, there are always these stories about ring speciation. Just the loss of ability to interbreed doesn't prove that salamanders came from fish.
I'm not saying they are. I'm saying
No, but that's what evolution says. And if the loss of ability to interbreed proves evolution, then it would prove that salamanders came from fish. It would prove that the general theory of evolution is true.
I kind of see what you're saying.
So you're not saying that ring speciation actually is evidence for real evolution.
I'm saying that through their changes,
Yeah.
how is that not evolution if they are changing genetically where they cannot mate?
Well,
Or produce
okay, the
offspring.
reason now look at like what I said, they're separated from each other.
Yes.
We don't get any new traits with new DNA that's better. What we've got is mutations that are endemic to each population, and their genome has dealt with it, either by repair mechanisms or by compensations or accommodations of some sort. But this group, if it's been thousands of years, maybe millennia, has got different mutations dealing with, and they've accommodated those. And these have got different ones over here. It's almost like languages in humans. I mean, you get some people off on a little island off the coast of England for 1,000 years, and they say things like Erin go brag. They're speaking Gaelic, and no one else on the planet can understand them because they've gotten off by themselves language-wise. They've also gotten off by themselves genetic-wise, not to the point where they can't interbreed, freckly-faced Irish people, but they still have their own mutations they're having to deal with in their population. Well, just the fact that two populations can separate for a long time and then their genomes can alter to the point where they're not compatible anymore or only marginally compatible, like mules are usually sterile, doesn't mean that this is evidence for Darwin's theory. It is genetic drift. You can even get bottleneck effects. You can get founder effect. You can get all these things that happen that change the genomes of both to the point, especially in something plastic genetically like salamanders. You could actually get it where they can't breed anymore. Even if they tried to breed, their DNA wouldn't recombine in fertilization. But that does not mean that it's an onward and upward thing. You may have created two groups that can't breed anymore, but that's like cutting a pie in half and saying you've made two brand new kinds of pie. You see, that's not a new species unless your only definition is they can breed with each other, and now that they can't, they're two new species. But like you pointed out yourself, their DNA really comes from the same spot. It's the copying errors that have accumulated over the many generations of separation that make their DNA incompatible. Now, some creatures can actually overcome this. Even with different chromosome counts. I mean, horses have lots of different numbers of chromosomes, and they seem to manage to interbreed just fine, whereas if that happened with humans, I don't think that our mechanisms do it. Does that help?
Yes,
Okay.
this is going to be our, I know you guys want to re-ask questions, but you guys can probably talk to him afterwards. This is going to be our last question because our room reservation runs out.
I got lucky, I'll just say that. A quick quote from a book called Freakish Luck I finished recently. Dr. Herbert Nilsson, director of the Botanical Institute at Lund University, Sweden, was honest enough to admit, my attempt to demonstrate evolution by experiment carried on for more than 40 years has completely failed. The idea of evolution rests purely on belief. In light of the topic, and I'm going to use archaeology as an example, okay? We have theories, right? And so we find a piece of hardened clay. We have two theories, one that it naturally occurred and one that it occurred by, or was created by an intelligence. Both theories, therefore, have
okay, so let's clarify for the audience. You're talking about an artifact, two artifacts that you find. One of them was maybe made by wind erosion, and it just looks like the Sphinx. The other one is so intricate that you're compelled to believe some humans carved this, right?
Yes. Yeah. So in light, okay, we find a hardened piece of clay. We have two possible causes, right? They both make predictions. Based upon those predictions, we apply that to what we find, and therefore we determine one to be true and one to be false. Correct? So if that's the case, would we not therefore apply that to biology? And if and that being, is that therefore non-scientific, or could you maybe clarify, for instance, Dembski calls it the design inference, and if it's an inference, we would still therefore determine one is true and one is false? Basically, I'm just looking for clarification in that area.
Well, when you see something and it has an intricacy to it, you might believe that the intricacy happened by some natural process and there was no mind, no body behind it. But the more intricacy you see, the more you just can't buy that it happened by itself. And our genome is a good example of that. Dembski and the design inference, we're talking intelligent design theory, we're talking information theory. I always like to say that the more information you actually see in DNA, for instance, the more you're sure some body is behind it. Let's say you're walking across the parking lot here on this hill on a nice day, and you got your girlfriend or your boyfriend with you, and girls like to look at clouds and imagine teddy bears and unicorns, so it's a romantic tip for you. But let's say we were looking up in the sky and you see what looks like an artifact in the sky. The letter T. Okay, it looks like a T, a little slanty, a little floaty in the clouds, and you don't have to think someone's up there with one of those little airplanes with the smoke going behind it like the Wicked Witch of the West, Surrender Dorothy. You don't think necessarily there's a skyrider, but you see a little slanty T. You might be able to convince somebody that the clouds did that. Look at that, isn't that cool? It kind of looks like a T looking at the clouds. What if it said THE? Maybe, maybe, maybe. Especially if it looked a little messy, you would go, no bodies behind that. This is just the clouds just did this. But what if it said the whole universe was in a hot, dense state? When nearly 14 billion years ago expansion started, the Earth began to cool, the autotrophs began to drool, the Neanderthals developed tools, we built the wall, we built the pyramids, math, science, history, unraveling the mystery that all started with the Big Bang, bang! Okay. No one could convince you that the clouds just floated and did that. You'd go, no, somebody's behind that. Someone is behind that, a mind with a capital M. Somebody did that. Now, just the letters in the right order for the jingle for a soap opera, I guess it is, a sitcom, that would be enough to convince you, that's enough information, that's enough intricacy to make you go, no, there's an intelligence behind this. But even the simplest genome of even the hypothesized first bacteria, the Archaeobacter we all came from, would be thousands of times more complicated than that. And to say that could just happen by itself without a mind behind it. And that's why I was saying to the other gentleman, information that is descriptive, that's prescriptive, that's a recipe, that's directions, it's instructions, never, we can't even imagine information like that coming from any place but a mind, because it never does. I've challenged college professors, and you can't come up with an example of information being generated from matter all by itself of this nature. I mean, tree rings tell a story, but that's just a knee-jerk reaction of the living thing interfacing with its environment. That's not actually telling you what to do. And crystals growing, the affinities of the atoms involved in the molecules of the crystal actually control that and regiment that. But where did that information come? That was instilled at the creation of those atoms, whether they happened by the Big Bang and nuclear fusion in supernova stars, or whether God made them. And what I'm trying to say is, yes. Well, it depends on how extreme it is. I mean, if the sitcom letters are enough to convince you there's a skywriter up there, that's enough information. Maybe the THE you'd buy, that happened by itself. The more information you see, the more intricacy you see, the more interdependent message that's actually there, the more you become certain there's a mind. And our genome with 3 billion base pairs, that's 6 billion nitrogenous bases lined up in a perfect order in 23 chromosomes, 6 feet, well, 2 meters of stuff. That's way over the top, enough to satisfy beyond the impossibility threshold that somebody did that. Now you could say, okay, well, evolution did it. And Michael Roos likes to say that the order, the design emerges as evolution creates. But what about the very first living thing where the first order happened by accident then, and there wasn't any guiding force, it was just chemicals floating in the ocean like we saw in the high school science textbook? That is a big, big pill to swallow. It's a lot to be expected to buy into. You can, but I just object to teaching it as the only way to think, and there aren't other ways. Does that help then? Yes, okay. We're going till what, 8:30?
That's actually it, because we got to be out of here by basically 8:30 so they can clean up. So let's thank Dr. Jackson one more time.